Julie, thank you very much. And I'd like to thank all of you for attending. I'm very much looking forward to this. I'm sure all of you are involved in one way or another on at-home instruction and learning. I am too with my college courses, so I know some of what you are going through. I want to thank Julie for her very generous introduction. I figured I'd give myself a quick introduction to all of you. Many of you know who I am, but lots of you probably don't. I've been working and researching and teaching at Brown University in Providence, Rhode Island for a long time. I'm a cell biologist. I work on biological membrane structure and function. I publish in journals like Cell and the Journal of Cell Biology. I've also written, as Julie was kind enough to mention, a number of popular books. These are basically aimed at a popular understanding of evolution and some of its implications. And also, together with a wonderful guy named Joseph Levine, who gave a webinar in this series on Monday, I'm co-author of a whole series of biology textbooks intended for high school students that have been published by Pearson. So a little bit of what I'm going to say will be in the context of that. Now, one of the interesting things about being both a textbook author and a college professor, I teach freshman introductory biology, among other things, is that you get to revisit the way in which the field has changed. Because one of the really cool things about science is it doesn't stand still. We keep having new things to add to what we learn. Now, way back in ancient history, when I was in college, of course, and even when I was in high school, I learned about the work of Gregor Mendel, the concept of the gene, dominant, recessive, alleles, and so forth. And you might say that was classical Mendelian genetics. And again, in the ancient past, when I graduated from high school, I thought I had a really good knowledge of genetics. Well, right about the time I was in high school, all of this began to be upended by what you might call the story of DNA, the birth of molecular biology. Now, this work was actually done in the 1950s, but it didn't really influence research until the late 50s and early 60s. And that's when I began to hear about it when I went into college. And of course, what we know, and the story that's in all the textbooks and lots of popular sources, is that two scientists, James Watson and Francis Crick, used X-ray data that had been produced by Rosalind Franklin to build a model that solved the structure of DNA. And an awful lot of what we have built on in the entire field of cell and molecular biology ever since has grown out of that discovery from these three remarkable scientists. Now, I thought, almost as a diversion, that I'd tell you about something that happened just a couple years ago. And it's a remarkable story. And I think you'd be interested in this whether you're a student or whether you were an educator. The very first written report of the double helix structure was written by Francis Crick, but he didn't write it for a journal or a granting agency. He wrote it for his little boy, Michael. Now, the picture you see here shows Francis Crick, one of the co-discoverers of the double helix structure, with his little boy, Michael. The letter was written when Michael was about 13 or 14, so this is a little bit later, but couldn't find a picture of Michael at that age. And the letter was kept by Michael until he put it up for auction a couple of years ago. And the actual letter went for $6 million. And you might wonder, what's in there that's so amazing? Well, on the very cover page, and you can see it right here, and the letter is available on the web, this scientist writes to his little boy, "My dear Michael, Jim Watson and I have probably made a most important discovery." Now, as you go through the letter, what you see here is Francis Crick's first drawing, first one he ever made, of the structure of DNA in order to communicate it to anybody else. And it shows a sugar phosphate backbone with bases sticking out to the side. Those bases are the A's, T's, G's, and C's of the genetic code. Later on in the letter, he tries to draw the double helix. And you can see the double helix right here. And you can see how he sketched the bases pointing to the inside. Just a remarkable thing to see. And then right here, you see his explanation of how DNA can replicate, how it can copy itself. Now, they didn't actually publish this until the replication scheme, until their second paper. Their very first paper really didn't mention it at all, but he wanted to make sure that his little son knew about it. So I have found it remarkable to realize that the very first written report of the double helical structure from DNA was signed "Lots of love, Daddy." And I think that, gives you sort of a human view of what the nature of scientific discovery is like. Now, one of the reasons I wanted to point this out to everyone is that National DNA Day is coming up in just a couple of days. In the area where I work, there are several high schools that have DNA festivals on these days, and I've been privileged to visit a lot of high schools and speak to them. Obviously, the schools closed everywhere. We're not having group celebrations of National DNA Day. That particular date in April is the anniversary of the publication of the double helix manuscript. I thought I'd mention that to you. And I thought those of you who are instructors, who are teachers, might want to dig up this letter, the copy of it on the internet. It is just remarkable, and it makes for very, very interesting reading. Now, when Joe Levine and I put together our latest textbook, we decided we were going to put letters to students in the front of the textbook. Now, I know that nobody ever reads these, but I wanted to point out part of the letter that I wrote for our last textbook. And that was, I wanted to tell students right now that they have been born, and you may not think this from the news today, but it's true. You have been born at just the right time. In all of human history, there's never been a moment like the present, a time when we stood so close to answering the most fundamental questions about the nature of life. That's an extraordinary thing to know. And students, you belong to the very first generation of students who can read the genome, almost like reading a book or a newspaper. Now, one example of this is the idea of gene therapy, which frankly used to be a fantasy. And that fantasy was, if someone had a genetic disorder, it might mean that they had a defective gene in cells throughout their body. Gene therapy would involve inserting a good copy of that gene and restoring cellular function. The initial attempts to do this, which took place about 20 years ago, were successful. So the young girl you see in this photo, Ashante Da Silva, is now 31. She was the very first patient ever in the history of the world to be treated with gene therapy. She had a disease called SCID, severe combined immunodeficiency. She was born without a functioning immune system. She had to live her life in a bubble when she was a little kid to keep from getting sick. Well, the reason her immune system wasn't working was they lacked a gene for an enzyme that's required for immune cells to develop. Well, it turns out her own cells were then taken out of her body, transformed with the gene for the enzyme, and put back in. And lo and behold, it worked. She came out of the bubble. She has a functioning immune system today, and she's been able to lead a normal life. It's a great success. So there is, in fact, and has been a cure for this genetic disorder. But I have to show you something. And again, this is from about 20 years ago. And this is a snapshot of a news article about this called "The Best of Times, the Worst of Times." Now, I told you what the best of times was. She was cured. What was the worst of times? Well, at about the same time, an experiment was taking place in the United States. And a young man named Jesse Gelsinger, who suffered from a particular genetic disorder, and you can see the name of it up there on the screen, volunteered for another experiment in gene therapy. Now, Jesse Gelsinger was getting along okay. He was having regular transfusions to replace an enzyme that was not present in his body. But he wanted to volunteer for a technique that might be able to cure young boys and girls who had a more severe version of this. So the idea was to take a healthy gene for this enzyme, put it into a modified virus, Viruses are really good at getting into human cells, as we all know too well today. And the idea was that this virus would be disabled, so it couldn't make him sick, but it would transfer the gene into the nuclei of his own cells, specifically his liver cells, because that's where that enzyme is made. They'd start making the enzyme and he'd be okay. Well, here's the sad part. Here are his parents, 10 days after the experiment, scattering his ashes. Jesse Gelsinger died, and he died because the physicians miscalculated the viral dose. His body went into shock, and he passed away. So it was, in fact, a great tragedy. This is a picture of Jesse in front of that very famous statue of Rocky, Sylvester Stallone, in front of the Philadelphia Museum of Art. He wanted to be a hero. He was a hero, but unfortunately, the experiment misfired and it cost him his life. So gene therapy is not straightforward. And I think it's fair to say that after Jesse Gelsinger was killed, literally, by an attempt to cure a genetic disorder, for more than a decade and a half, the dream of curing genetic disorders this way has simply been on hold. Many researchers have been, frankly, afraid to try it. And instead, this has been displaced by a new field, stem cell research, which I'm sure you've heard a lot about, and stem cell therapy. Now, I'm a cell biologist, so I find this field very, very exciting. And I think there are fundamental breakthroughs being made that are going to have importance in the clinic. They're going to help people. But now, and this is what I wanted to talk about today, the prospect for genuine gene therapy is back. It is, in fact, already being done. Now, the interesting thing about this is that it has come about initially in a most unexpected way. What you see on the screen right now, on the left, is an electron micrograph of a mitochondrion. These are sometimes called the powerhouses of the cell. This is where the Krebs cycle takes place. This is where electron transport and oxidative phosphorylation take place. Look into the respiration chapter of your textbook and you'll find out all about it. This particular mitochondrion is probably in the process of dividing into two. Now, why are mitochondria a target for gene therapy? The electron micrograph on the right shows why. That is the DNA molecule of a mitochondria. It turns out mitochondria have their own DNA. They're separate from the nucleus. So in effect, they have their own genome. And you can actually see the circular nature of this particular DNA molecule in the electron microscope. Now, mitochondrial DNA molecules are small. They're circular. Human mitochondrial DNA has 17,562 base pairs. 17,000 is not much when you consider that there's 3 billion base pairs in the haploid genome inside the nucleus. And you can see the labels or the names of some of the genes in human mitochondrial DNA right here. Now, we've known about mitochondrial DNA for, oh my goodness, more than 35, maybe even 40 years. But no one thought that it might be related to genetic disorders until this guy. And this is Doug Wallace, who is a clinical physician at Emory University in Atlanta. Dr. Wallace's specialty was treating genetic disorders. And he came across a family who presented themselves to him as patients, in which there was a genetic disorder over many generations, which is called Leber's hereditary optic neuropathy. And people with this disorder, usually in late adolescence, began to lose their eyesight. Sometimes they just lost a lot of visual acuity, so things look fuzzy instead of sharp. And sometimes they went totally blind. So there were a bunch of things about this that puzzled Dr. Wallace. One was, first of all, it starts in late adolescence. It's not a childhood disease. The second thing I've already mentioned, which is variable severity. Sometimes you go blind. Sometimes it's not so bad, you just lose some sharpness in vision. But the most puzzling thing of all was maternal inheritance. Now, this is really different from sex-linked inheritance that you might have read about in a genetics chapter. Maternal inheritance means that a man with Leber's would pass it along to his children. So if you were the son or the daughter of a man with Leber's optic neuropathy, your risk of getting it was 0. On the other hand, if you were the son or daughter of a woman, a mother with Leber's, your chances of getting it were very good. So the disease could be passed along to either male or female children, but only the mother could pass it along. Now, Wallace thought about that. That was completely different from Gregor Mendel's patterns of inheritance. It's not sex linkage, it's something really weird. And he came to the conclusion that maybe this resulted from a defect in mitochondrial DNA. And here's the reason why. Each of us got 23 chromosomes from mom, and we got 23 chromosomes from dad. But we got all of our mitochondria from mom, because our mitochondria come from the cytoplasm of the egg, which is fertilized to form the single cell that gave rise to each of us. So Doug Wallace called up a friend at the research arm at Emory and asked them to help him analyze mitochondrial DNA in this family. And what he discovered was his hunch was right. They had a mutation. There was a single base pair changed at nucleotide 11,778 of the human mitochondrial genome, and it changed an arginine to a histidine of amino acid number 340 and one particular protein in the electron transport chain. Now, such a small mutation, how could it cause blindness? Here's the deal. What that change meant was that that particular enzyme in the mitochondrial electron transport chain was just a little bit less efficient in making ATP. Well, it turns out the cells of the optic nerve have some of the highest demands for ATP in the whole body. So when these people had a mutation that made that less efficient, many of the cells in the optic nerve couldn't keep up with the demand for ATP. And by the time they had reached adolescence, their eyes and their bodies had stopped growing. They'd occasionally hit peak demand for ATP in one of these cells. Their mitochondria couldn't survive it, and the cell would die. That would increase the load on the other cells in the optic nerve, and little by little, more and more of the optic nerve would be destroyed, sometimes resulting in total blindness. Now, I've always felt that Doug Wallace deserved the Nobel Prize for this because he single-handedly convinced people that the mitochondrial genome was important. And it turns out we know that it is. Once he opened the door, we now realized that there are a whole series of genetic disorders based in the mitochondria. And right now, of the 37 human mitochondrial genes, we know about genetic disorders associated with 30 of them. And I'll bet you when the research is completely finished, it'll be 37 out of 37. And I want to give you one quick example of this. And it's an example that's familiar to me because I've actually met this guy. This is Rocco Baldelli. Brown University is in Providence, Rhode Island. He grew up in Woonsocket, Rhode Island, which is right next to it. A relative of his is actually my wife's best friend. Rocco was maybe the best baseball player to come out of Rhode Island in decades. He played for Bishop Hendrick in high school. He batted, if you know baseball, think about this. He batted .531 in his senior year in high school. He was picked number 6 in the major league draft. He signed with Tampa Bay Rays for a whole bunch of money. He was the minor league player of the year in 2002. He almost won Rookie of the Year the next year, but then suddenly he retired in 2006. And here you see a news article about his retirement. Illness forces Tampa Bay outfielder to retire. This guy could play. He could throw, he could run, he could hit, he could field. He just had everything. But he quickly found as you played 162 games a year, he simply couldn't recover. He was fatigued more and more and his performance suffered. It turns out that he was indeed suffering from a genetic disorder. That's the name of the disorder, mitochondrial channelopathy. And I won't go into details, but basically mitochondria have to import a lot of their proteins from the rest of the cell, from the cytoplasm. These proteins have to go through a channel to get into the mitochondria. He had a genetic disorder in the gene for the channel protein, so it didn't work so well. So his mitochondria were quite literally not firing on all cylinders. Now, I don't want you to feel bad for Rocco Baldelli, because even though he had to retire as a player, people realized he was a really smart baseball guy. And he coached for a number of years. And in 2018, he was named the manager of the Minnesota Twins. And if you follow baseball, you know that last year he was the American League Manager of the Year. So this is fortunately not a fatal genetic disorder, but on the other hand, it's serious, but he was able to survive it. He's going to lead a great life, and obviously he's one heck of a manager. So I'm very happy for Rocco. Now, one of the reasons I bring this up, and you'll see what I mean in terms of genetic engineering, is something that I wrote a case study about in the latest edition of our textbook. And the case study is entitled, "What's Happening to Me?" And I talked to a couple physicians who had treated people with Leber's, and from their descriptions came up with this little graphic designed to show how as Leber's advances, the visual field gets blurrier and blurrier and blurrier. And this was an introduction to the role of the mitochondrial genome. On the very same case study, I wrote a feature called Technology on the Case. And the subtitle is Stopping Leber's Hereditary Optic Neuropathy Before It Starts. How could we fix this genetic disorder? And there you can see it, stopping it before it starts. Remember that it's caused by a defect in mitochondrial DNA. What would happen if we were able to replace mitochondrial DNA? So, for example, let's suppose that Mr. Baldelli had a sister who also had the same problem he did, because she also inherited these abnormal mitochondria from Mrs. Baldelli, their mother. Well, how could we fix that? Remember, any of the eggs that she produced would contain the defective mitochondrial genome. However, let's suppose that we took the nucleus out of a cell by using in vitro fertilization and put it into the cytoplasm of some other woman's egg that had normal mitochondria. So the idea, and you can see it in the little red box here, is to take the maternal nuclear genome, because the nuclear genome is okay, and insert it into an egg cell, an oocyte, from a woman with healthy mitochondria. Then what would happen, and here you can see it now, is you could then take this cell in which the nuclear genome was the same as the mom, but the cytoplasmic genome, mitochondria, had replaced it, allow the father's sperm to fertilize it, in vitro fertilization, which happens all the time. And if it went well, you would then develop an embryo with the nuclear genome of mom and dad, but the cytoplasmic genome, the mitochondrial genome of that donor. And that way there would be no mitochondrial disease. It's a kind of human genetic engineering that could eliminate a mitochondrial genetic disorder. Now, in a sense, children born this way have three parents, because they have mom who gave the egg nucleus, we have dad who gave the sperm nucleus, and then the third parent gave the mitochondria when she donated an egg. And in fact, we have three parent babies. Once it became apparent that this technique could actually work, there are a lot of discussions about it in advanced countries like Great Britain, the United States, Germany, and so forth, as to whether or not this should be allowed to go forward. Well, it has gone forward. And in fact, almost 3 years ago, or more than 3 years ago, the very first baby with DNA from three parents was born. This technique is actually banned in the United States. So the transfer was done in a third country. The baby was actually born in the United States. And here you see the physician, Dr. John Zhang, in New York City, holding the baby. And this baby had the mitochondrial genome of a third parent. It turns out this has now been done many, many times. And the question that we all have to ask at this point is this an ethical procedure? And what I mean by that is it okay to do this? And you might wonder, how could there be any ethical questions if you're eliminating a genetic disease? I take that point. But let's suppose, just for the sake of argument, and I always like to personalize things, um, I was a competitive swimmer in high school and college, and I was quite successful, but I was never Olympic material. Believe me, never. Let's suppose my wife and I wanted to have children and we wanted them to be Olympic material. And we heard that, let's say, Michael Phelps' sister, same mitochondria as Michael Phelps, was willing to donate her egg cytoplasm. Just imagine how good Michael Phelps' mitochondria must be that they enabled him to win 30 Olympic medals. Would it be okay? I don't have any genetic defects in my mitochondria, or nor does my wife that I know of. Would it be okay to swap out mitochondria for the mitochondria of a star athlete? Well, it's something to think about. And in fact, there are other ethical questions with this as well. But nonetheless, this kind of genetic engineering, producing 3 parent babies, is already feasible and is already being done, and is in a sense the first kind of successful human genetic engineering in quite a while. Now, at this point, I don't know if there are any questions, but I'd like to pause for a second, and Julie can tell me if she's gathered any questions, or let you know that if you have any questions, there is a way to ask them. Julie?
So actually, right now, we don't have any questions in the chat box, but if anyone would like to type it in, I see there's a couple people with their hand raised, but I don't see an actual question yet typed in. Oh, got the first one. By bringing this up, are you a proponent of
this? That's a good question. I try to stick to the science and let people know that there are really serious ethical issues here. I think for the most part, I don't see any objection that would concern me about using this technique to alleviate human suffering and disease. So in terms of eliminating gene defects of the sort that I've talked about, especially something like Leber's, which causes blindness, if I was faced with this issue in my own family, I would want to go ahead and see if this genetic disorder could be replaced, could be fixed. In terms of human enhancement, making a faster runner, a faster swimmer, or something along those lines, there I'd sort of scratch my head and say, no, I'm not so sure about that. It's one thing to try to alleviate disease, and it's another thing to try to hack the human genetic system to produce a so-called superior human being. So I'm giving you a yes and a no.
Another question we have, and I think you kind of did this in your answer, so I don't know if that's fulfilled, but it says, can you please describe the ethics in relation to genetic engineering?
Well, I've already mentioned that a little bit, but I'll mention one more, and that is a US panel in the United States, charged with deciding if this was ethical or not, said it should be okay to go ahead. They would approve treatments like this for genetic disorders, but they would only approve them for procedures that would produce male babies. And at first you might think, whoa, what's going on here? But I want you to think about this. The reason they suggested that is males don't pass their mitochondria along to their children. So if something went really, really wrong, it would end with that one person. Females, of course, do pass their mitochondria off to their offspring. So that's why this panel said, well, we'll only fix it in male babies, not female ones. That opens up a whole series of other ethical questions in terms of who should get this sort of treatment and who should not. So thanks for both those questions. I'm going to move on, and I'll be able to take some more questions later. So here's what I want to move on to. In 1993, more than 20 years ago, a paper reports something: frequent repetitive DNA motifs in bacteria of unknown function. It was a very obscure paper at first, and almost 20 years went by before the importance of this discovery was realized. Now, what these people had discovered were a series of palindromic repeats in the DNA of a particular bacteria. Now, what you might ask, the palindromic repeats are represented in my diagram by little double arrows. What's a palindromic repeat? A palindrome is a sequence of letters that reads the same way in one direction as it does in the other. You can apply that to words in the English language. In DNA, here's what we mean. Look at the top strand of DNA. It reads TTAGCAC. Now look at the bottom strand. You'll see that it reads exactly the same way on the opposite strand. That's what a palindrome means in DNA. And it was very surprising for these investigators to discover all these palindromes with the same sequence, I might add, repeating. Now, in between them were non-repeating sequences of what at first looked like gobbledygook. Nobody was sure what that was. But basically, the way in which they described this genetic structure, which was very strange, they called them clustered, regularly interspersed short palindromic repeats. The abbreviation for that is CRISPR, and you may have heard of CRISPR. Now, what is CRISPR really in the bacteria? It's actually a defense against viruses. So what happens is those palindromes separate snippets of viral DNA. If a virus infected a cell and the cell survived it, the cell would keep a little souvenir of the viral DNA behind just in case the same virus showed up later. If it did, they would then transcribe RNA from those non-repeating frequencies, line them up with an enzyme. They'd create an RNA destruction machine or DNA destruction machine that would find the viral DNA, cut it into pieces, and inactivate it. Almost an immune system. So what the CRISPR system does is it cuts DNA at very specific places, specified by RNA made from these little pieces of viral DNA. So when this was realized, it was cool, it was interesting, but nobody thought the practical value of it until two researchers got together at a conference and started to talk about it. Those two researchers were Jennifer Doudna from Cal Berkeley and Emmanuel Charpentier from France. They realized this could be made into a tool to edit DNA. Another scientist, Feng Zheng at MIT, modified it so it could work in mammalian cells, including human cells. This is a revolution in genetic engineering. Now, I'm not a betting person, but if I had to bet on who's going to Stockholm this year for the Nobel Prize, these are the three people I would bet on. So hold the phone until September and let's see if I'm right. Now, how does CRISPR work? The system consists of a piece of RNA called guide RNA and an enzyme called Cas9. And this diagram shows the guide RNA has a double-stranded loop that binds to the enzyme. And then it has a single-stranded region, which is target specific. What the single-stranded region does is it base pairs with a specific sequence of DNA. Depending on the base sequence there in the RNA, it can find any piece of DNA. And the Cas9 enzyme will then go snip, snip, and make a double-stranded cut. Now that's how it inactivates viral DNA in the bacterium. But here's what you can do with it in, for example, a human cell. And this is why it's a big deal. The first thing that it can do is by adjusting the sequence of the guide RNA, you can cut and disrupt a gene by causing a targeted double-stranded break. And that's what you see here inside the red box. If you target that to a particular gene and you go snip, snip, you create a double-stranded break in the gene. The cell has a repair mechanism that cements the break back together. But when it does so, it throws in random bases. Those random bases almost always render a gene non-functional. So it's a way basically to knock out a gene. The other thing you can do with CRISPR is if you also include a new piece of DNA whose ends match the DNA on either side of the cut, there's a system inside the cell, it's called homologous recombination, that can take that piece and cement it in thereby changing the DNA sequence exactly as you want it to be changed. We have never been able to do that before. That is literally revolutionary. In the last edition of our book, I thought it was very important to explain to students how this works and how you can literally edit DNA with it. We now have in the laboratory the capacity to take any DNA sequence, let's say AAA, TTT, and we can change it if we want to AAA, CCC, and we can do it quite easily and precisely with CRISPR. Now, it turns out, and this slide simply shows some of the techniques by which you do it, it turns out that CRISPR is actually simple. To make CRISPR work in a cell, all you have to do is, first of all, insert into the cell a gene for the guide RNA that is complementary to the DNA you want to modify, then a gene for the Cas9 protein, and perhaps if you want to replace it rather than simply disrupt it, a piece of DNA to go into and patch the sequence. It's actually very simple. It's not expensive to do CRISPR experiments, and this technique has sort of taken over by storm in terms of genetic engineering. Now, is it useful for the human genome? The answer is yeah. Let's take an example, and the example would be sickle cell anemia, which is a hereditary genetic disorder in this country, and it causes red blood cells to sickle, clog the bloodstream, all sorts of problems. In this country, it's mostly found in Americans of African ancestry. Around the world, it's found in people from anywhere in the world where there's a high incidence of malaria, not just in Africa, but also in the Middle East and in the Indian subcontinent. And the reason for that is having sickle cell basically serves basically to make you resistant to malaria. But if you have 2 copies of the sickle cell allele, you can be very, very sick. So we've struggled for a long time on ways in which the sickle cell gene, sickle cell allele could be cured. The initial defect in sickle cell is in the genes that code for part of the hemoglobin protein, specifically one called beta globin. There's a single base change that causes the sickling of hemoglobin. Now, however, there is a way to use CRISPR potentially to actually cure sickle cell. And the idea is to bypass the adult genes that produce that part of the hemoglobin protein and instead activate fetal genes, which are right there on the chromosome right next to it. These are genes that produce hemoglobin when you were a fetus. The hemoglobin works fine. It's not affected by sickle cell. The hemoglobin we make as a fetus has a slightly higher affinity for oxygen. That's why we have multiple genes. So the idea is to activate these genes that are silent in an adult to produce fetal hemoglobin, which presumably would not sickle. Well, the young woman you see here, Victoria Gray, bravely, I think, volunteered for the very first experiment attempting to cure this. And her own bone marrow cells were modified using CRISPR. Then they're put back in her body. And lo and behold, the news has been good month after month, producing functional hemoglobin instead of sickle cell. This is a genuine cure. It's an extraordinary thing. Now, thinking about this going forward, it's important to point out that Victoria's treatment only affected her bone marrow cells, which produce red blood cells. It would not be passed along to her children in the future. However, is it possible that we could also use CRISPR to make permanent, heritable changes in human DNA? And the answer to that is yes. And that's one of the reasons why, for all of the promise of the CRISPR technique, this is something to think about and to think about now. Just a couple of years ago, a scientist tried to edit out a dangerous mutation from genes in human embryos, a mutation that would actually cause a fatal genetic defect. They used CRISPR to do it. This is the particular gene that they attacked, and the experiment was successful. Now, these embryos were structured in a way that they could never be brought to term. So the science, in other words, they'd never give rise to a baby. But the scientists did this to show that it was possible to edit genes in an early human embryo and fix a genetic defect. When that happened, a number of scientists, including one of my former students, Fedja Ornoff, wrote a number of op-ed pieces saying, don't do this yet. We don't know all of the implications of editing the human germline, of causing permanent changes in the human genome. Do we really want to be responsible for producing designer babies whose DNA has been edited? There's good reason to hold back on this, but at least one scientist did not. And a couple of years ago, a Chinese scientist announced that he had done this, and he had edited the DNA of two little girls in an in vitro fertilization experiment. And basically, this was a serious issue. It upset an awful lot of ethicists, and it even upset the Chinese government, who charged and convicted this scientist of doing unauthorized experiments. Why were people so upset? Well, he was quick to say that the gene that he edited was one for a protein called CCR5. Now, the reason he edited this gene to change the protein, was because this is the entry point for HIV, the virus that causes AIDS into immune cells. So he had said, when these two little girls grow up, they will never catch AIDS because the AIDS virus can't get in their cells. That's true. I do think there are better ways to avoid getting AIDS than having your DNA tinkered with by this way. But here's the reason that a number of people were upset. There are people alive today who have mutations in this gene. That's why he went for CCR5. These people have much higher incidences of type 1 diabetes. They are more susceptible to hepatitis virus, to the West Nile virus, and also to the flu. So at some point, when these little girls are old enough to understand, somebody's going to have to tell them that before they were born and without their consent, a scientist messed with their DNA, and this could have potential health effects. That's a serious issue. Having said that, think of a really serious genetic disorder like muscular dystrophy. Could muscular dystrophy be cured via CRISPR? Well, there is an animal model of muscular dystrophy. There is a line of beagles that have a very similar problem. And it turns out, what I'm showing you are slides from a paper showing that, in fact, a version of Duchenne muscular dystrophy in dogs can be cured by CRISPR. So do we want to go ahead and do that with human beings? That is a difficult question. And that's something that we all have to talk about before we move forward with this. There's another thing I wanted to talk about with respect to CRISPR. CRISPR is already being modified, already being used to modify the plants that we use as food sources. And as you can see here, the interesting thing about CRISPR is these genetic changes can be so subtle, you can't tell if these are the result of an ordinary mutant or a genetic manipulation. And in fact, there are at least two CRISPR foods already on the market. One is a genetically engineered mushroom. If you've ever cooked with white mushrooms, you might know that if you slice them up and leave them around for a couple days, they get kind of brown and slimy. Well, that color in the slime comes from an enzyme that reacts with oxygen. What a biotech company did was to use CRISPR to knock out the enzyme. And as a result, these mushrooms stay white and crisp much longer. The very, very same technique was used by another biotech company to inactivate a very similar enzyme that causes apples to go brown after they're exposed to the air. This is called the Arctic apple, and it's actually being marketed right now. So these apples basically will stay crisp and colorful for a very, very long time. They are not marketed as non-browning apples. And there's also some hope that CRISPR-driven genetic modification can save crops which are, like oranges, endangered by diseases like citrus greening. So CRISPR-modified foods are coming to a grocery store near you very, very soon. I want to give you one other surprising example of the efficacy of CRISPR. I'm sure you all know that there is a shortage of human organs that is needed for life-saving transplants. I have checked the organ donor box on my own driver's license, and I hope that many of you have as too. But there's still a shortage. And a number of physicians have said, you know, we might be able to get organs from pigs for use in human transplants. And the reason, before you laugh, is that many pig organs are exactly the right size and they function in the same way as our own liver, kidney, and so forth. But there is a concern, and that is pig cells are home to a slew of retroviruses, viruses that are actually in the pig nucleus. These are called, and please don't laugh, PERVs, P-E-R-V-S, for porcine endogenous retroviruses. These would be dangerous. I wouldn't want a kidney transplanted into me that had these potential viruses inside it that might make me sick. So the solution, believe it or not, turns out to be CRISPR pigs. A number of scientists have figured out a way to find the DNA sequences in these retroviruses and use CRISPR to inactivate them so that none of these viruses are active. And once again, CRISPR might just solve the problem in this particular case of making pig organs safe to transplant into humans. Just something to think about. Finally, and this is the last example I want to do, name the animal that kills more people, mostly children, every year than any other predator. It might surprise you. That's the animal. It's the mosquito. Specifically, it's the mosquito that carries malaria. That is a dangerous animal. What is malaria? Malaria is a parasitic disease. It's caused by a protozoan, and it's spread by the bite of this mosquito, native to Africa, by the way. And when it bites an individual, it sucks in some of the blood. If the blood has the malaria parasite, it grows for a while inside the mosquito, and the mosquito can pass it along to the very next person. The malaria parasite, this is really ugly to see, grows inside red blood cells until it eats up the red blood cell from the inside and causes it to burst, producing fever and chills and anemia. More than 1 million children are killed every year by malaria. This is a serious and enduring disease. Wouldn't it be great if we could get rid of that mosquito? So how could you do it? Maybe by inserting a gene into mosquitoes that makes it impossible for them to reproduce. Then you release these genetically modified mosquitoes into the wild. Sounds like a good idea. The problem is genetics, and that is Mendelian inheritance. Even if you introduced an altered gene, as you can see in the bottom, it really wouldn't spread. Only a small fraction of the offspring would have this altered gene. So how could you tweak this to make it work? The answer is use CRISPR to create something called a gene drive. Now, the technical details are pretty deep, but I want to show you basically what it means. A gene drive means you put the gene for the CRISPR system into one chromosome. What that gene is capable of doing is engineering CRISPR so that it attacks the same region in the homologous chromosome. Remember, we all have two sets. And basically, the gene replicates itself. And in so doing, it basically produces copies of itself in the other half of the genome. What that means, basically, is this gene will spread through the population. And if it includes a DNA sequence that prevents females from laying eggs, but doesn't prevent the male offspring from spreading the gene, what will happen is this gene will spread throughout the mosquito population until the population's egg laying capacity goes to 0 and the mosquitoes are eliminated. So what CRISPR does is to cut the other chromosome and then inserts a copy of itself plus the design gene. Does this really work? Whoa, excuse me, ladies and gentlemen, I just had a crash of my slide program. Hopefully I can bring this up again. I apologize for this. You're getting a good look at my desktop there. So let me go to the slide that I was about to show. So the question is, does this really work? Oh my goodness, it's crashing again. I may have a problem with that particular slide. So I tell you what I'm going to do. I'm going to try to skip it. Let's try one more time. And I'm going to skip that particular slide. I don't know if there's a problem with it or not, but it turns out this does indeed work. Now, you could use this for all sorts of reasons. Imagine how great it would be to eliminate mosquitoes spreading disease. You could eliminate invasive organisms. You could alter disease organisms that attack crops. But there's a number of problems. Organisms could involve resistance to it. Once a gene drive is released, it could get out of control. It might spread from harmful species to helpful ones. Imagine if we tried to kill mosquitoes and instead we killed honeybees. And the CRISPR modification could mutate to spread different genes through it. So we can alter species with a gene drive. We know this works, but should we? And that's the sort of ethical question that we have to consider. Gene editing could eliminate mosquitoes, but is it a good idea? Is the technology too dangerous to unleash? Now I'm going to skip over the very, very last thing I wanted to mention because I've begun to run out of time. This has to do with gene analysis kits like 23andMe. 23andMe is fun. You can learn a lot about yourself and your own genome, but it turns out you can also use 23andMe, and police can use it as well, to track down suspects in a crime. And there was a very famous case in California in which an individual called the Golden State Killer was identified from the 23andMe samples, or the genetic samples of distant relatives. So something interesting is going on. Now I'm near the end of the hour. I do apologize for that crash, but I wanted to make one more point. In talking about these particular issues, what I've done is I've written up 5 case studies. And these deal with the ethical issues that some of you have already asked about. And these include cases like 3-parent babies, CRISPR GMOs, gene therapy, pig organs, and CRISPR gene drive. These could be used individually if you're a student, just to think about. If you're a teacher, an instructor, you can download these and you could use these as lessons for your class. In the CRISPR one, for example, what the case study does is to explain the technique, to explore the experimental studies, to get students to research the risks and benefits, and finally to debate, engage, and allow people to work towards a conclusion. All of these are available to be downloaded. I'm sure Julie can explain how you can do this, but I'm very happy to share them with you and with your students. Why should we do this? The world is changing. We have to understand the technologies that genetics are giving us. We have to be able to ask forward-looking questions. What are the long-term effects? What are the risks and rewards? And finally, and as every year goes by that I work in science, I realize that science is going to continue to refuse to stand still. So all of us have to prepare for a lifetime of scientific change. And I think grappling with these questions right now in terms of ethics and practicality will help all of us to do that. Finally, in the event you'd like to get in touch with me, I invite you to do so. I have a very simple email address, which you can see right there. It is simply my name and my institution, and I frequently tweet, I should say, on scientific issues, and my Twitter handle is Kenneth R. Miller, as you can see right there. That's the conclusion of my presentation, and if questions have accumulated, I'd be very, very happy to try to answer them. And in the meantime, thank all of you for attending today. I have enjoyed this.
Thank you so much, Ken. We've been getting a lot of questions and a lot of comments. People are enjoying your webinar very, very much. If you have additional questions, please continue to ask them in the question box. I will go through and try to ask some of them. I know we have a lot, but Ken did post his email very generously too. So if we don't get to you, maybe you can reach him that way. Let's see. All right. Like I said, there's so many. Oh, here's one that's related to COVID-19, so I'm going to throw that one out there first. What possible applications could CRISPR have on viruses such as COVID-19?
Well, the very first thing I can tell you about COVID-19 is there are a number of laboratories that are developing rapid tests for the virus that use CRISPR. And the way in which these rapid tests will work is that you program in the known RNA sequence from the virus into CRISPR itself. So that little single strand in CRISPR can recognize the COVID-19 sequence. It then makes that cut that we've been talking about. And the open ends of the cut are then tagged with fluorescent bases. So very, very quickly by another enzyme. So very, very quickly you produce a fluorescent signal that can be read in a matter of minutes that tells you that the virus is there. So that is the one use of CRISPR that I'm aware of that is proceeding in very, very rapid fashion to try to improve our testing. We can do similar things with CRISPR potentially to detect the production of antibodies against CRISPR as well. I'm not up on those in terms of how far along they are. In terms of a therapy against the virus enters human cells by attaching to a particular protein. I suppose that you could use CRISPR to modify that human protein, and you could breed a line of genetically modified humans that had a modified version of the protein that the virus couldn't get to. You actually wouldn't want to do that because that protein on human cells is used as a signaling device for a whole series of physiological systems. So I don't see CRISPR as a way to modify human cells to make us resistant, but I do see it as a way basically to develop another rapid testing technique for the virus.
Okay. What do you suppose the change to the ecosystem would be by effectively eliminating even one mosquito species?
That's a really good question because I think of mosquitoes as a pest. I think most people here think of mosquitoes as a pest. But I was reminded literally yesterday morning, my wife and I live on a very small farm in Massachusetts, and yesterday was the day that our barn swallows came back. We have in any given year as many as 25 or 30 barn swallow nests in the rafters in our barn. Now we're really happy to have them. And one of the reasons we like them is, first of all, they reproduce, so we always have a ton of cute little barn swallow babies. Once they're ready to leave the nest, they fly all around our barn, and you can see them on humid evenings flying around with their mouths open, catching mosquitoes. So it turns out, as annoying as mosquitoes are, they are actually a major food source for these birds and for a number of other species. If you were to completely eliminate mosquitoes, you'd be causing problems going down the food chain, or up the food chain, I should say, for species that potentially feed on them. It's very difficult to imagine an ecological situation where you can completely eliminate one very common species and not have a cascading effect on others. So I think there are ecological concerns before you go into these, into do these experiments. And my barn swallows are just one example of that.
Okay. Instead of using CRISPR in humans, do you think that using CRISPR in pigs will make xenotransplantations common?
It's a good question as to whether it will make it common. It certainly is one big step on the way to making it feasible. I personally believe that the solution to organ transplantation is to work much, much harder on getting people to agree to become organ donors. I think that's a very important cause. I've made it clear to my family that if I'm ever in a traffic accident or something like that and I die, I have no use for any of my organs. And if they can be transplanted into anybody to help them to save their lives, please take those organs out. All too few people have arranged for organ donation, and that's one of the problems. That's the reason why people are thinking of xenotransplantation in the first place. When I've spoken about this in the past, I should tell you that I've had a number of people from different religious groups whose religions include prohibitions against the eating of pork, saying they really don't want any pig organs inserted into their bodies. And I certainly and completely understand that. So I think in terms of xenotransplantation, we already use some forms of xenotransplantation to replace heart valves and so forth. This might make it more possible, I don't really know if it will ever become common. And I actually know a couple of transplant surgeons who think this is a nice theoretical possibility, but they don't really want to get involved in it, certainly not at this point.
All right. Is this technique capable of alleviating all genetic diseases, or is it just associated with mitochondrial genetic diseases?
The mitochondrial transplant technique, the three-parent baby technique, there are only, as you saw, 37 genes in the mitochondria. So the only diseases you're going to be able to treat are ones that are in that tiny mitochondrial genome. The nucleus has anywhere from 20 to 22,000 genes. That's a much larger number. And the three-parent baby technique, the mitochondrial transplant technique, it just can't address those.
Okay. How is CRISPR different from previous methods of using restriction enzymes to create recombinant DNA in bacteria to produce human insulin?
That's a very good question. The big difference is that CRISPR is much, much, much more precise. CRISPR can go into an entire cell, a mammalian cell that might have 6 billion base pairs of DNA, and it is specific enough that it can find one individual unique sequence in those 6 billion base pairs, cut it, and then replace it with an edited sequence. We have never been able to do anything like that before. Restriction enzymes recognize specific base sequences that are anywhere from 4 to 8 bases in length. And what that means is, let's take the bacterium E. coli as an example. E. coli has a much smaller genome than we do, only about 4 million, not billion, but 4 million base pairs. When you treat it with an enzyme like HIPA2 or EcoR1, those enzymes cut at very specific 6 base pair sequences, but there are anywhere from 100 to 250 of them per cell. So the restriction enzyme doesn't cut in just one place. It cuts in many cases in hundreds of places. You then have to work very hard to find exactly the right fragment that you want. And when you use a bacterium to make human insulin, which is the example in your question, what you're doing is you're taking the human insulin gene and you're splicing it into a restriction site in a bacterial plasmid and putting it back in the bacteria. So you are taking a gene from one organism, humans, and putting it into another one, E. coli. The cool thing, as useful as that is, the cool thing about CRISPR is it doesn't involve taking a gene from one organism and putting it into another. It involves taking a gene in one organism and editing it. In other words, changing the gene right on the spot. So it's sort of the difference between using a word processor to correct a spelling mistake and pasting in an entire piece of paper from another typewriter to change a whole paragraph. So CRISPR is much more precise. It can work directly in a living cell, and it doesn't require the addition of so-called foreign DNA. That's the big difference.
All right, I'm thinking we do maybe one more question, and then Ken, I could send you the list because we still have about 25 or so questions on here. I want to be
That would be great. That would be great. I'll try to answer all those questions. And as you already know, I've invited people to go ahead and send me email. It'll be a pleasure. Go ahead.
Okay. This one's interesting. We know that COVID-19 is naturally occurring zoonoses that jumped to people last year, but many people think it was created in a lab. How do we disprove that hypothesis? Could we use CRISPR to make new viruses already, or is that still really far off?
CRISPR is a DNA editing technique, and therefore it is entirely possible to use CRISPR to change any DNA sequence. How do we know where this virus came from? There's a very, very good study that was published in the journal Nature, analyzing the genome of this particular virus. It is sufficiently similar to viruses found in other mammals, notably in bats, that it's very, very clear that it evolved from a natural population and was not genetically engineered in the laboratory. Because the tools that we have to genetically engineered viruses would leave certain marks in the DNA. None of those marks are in the COVID-19 virus. So please don't believe conspiracy theorists who say that the COVID-19 was cooked up by the CIA or it came out of some secret germ warfare laboratory. It very much looks exactly like a mutant form of a natural virus that underwent something we call a host range mutation. Could you use CRISPR to genetically engineer a virus? I suppose you could, but even that would leave certain marks that you'd be able to tell. So I don't think that we have to fear CRISPR in that respect, in terms of the mad scientist concern. I do think we have to sort of scratch our heads and think about how to use wisely the power, the control over living organisms that CRISPR gives us. That's the challenge we're going to face in the decades ahead.
Excellent. Thank you so much, Ken, today for joining us. And those of you that joined the presentation, we really hope you had a nice time. And we will be posting the recording of this probably tomorrow up on the blog. We also have 2 more biology webinars coming up at the same time, tomorrow and Thursday. And we hope you have the ability to join those as well. And you can always follow us on social media or check out the Pearson blog for more information. Those of you that wrote what blog in the chat, I will paste it in right now so you have the link. Thank you and have a wonderful day.
Thank you.